Thanks, everyone. Morgan, appreciate you. The floor is yours.
Hi, everyone. Thank you for having me here today. It's quite a privilege to present. Thank you for the invite, and I hope that I can provide something useful to you through this talk. So today I'll be discussing field sampling strategies in unstructured agricultural settings. So unlike structured experimental trials, field sampling in agricultural environments comes with unique challenges. So in this talk, we'll explore different sampling methods, how to determine appropriate sampling sizes, and the best practices for managing spatial variability. So let's dive in. So I want to start off by showing you some pictures of these beautiful grasslands. And you can see the picture on the top left-hand side, that's actually not a grassland. That picture is taken from subantarctic Marion Island. But the rest of the pictures are from grasslands in the Drakensberg. And as you walk through these grasslands in mountainous environments, one thing that you'll notice very quickly, and of course on Marion Island as well, is you have very changing conditions over small spatial scales. So the conditions change very quickly. You go from steep slopes to shallow slopes, and you have unique landforms. So you have rocky areas to deeper soils. And through my research, I've worked in a variety of these landscapes, which I will refer to as being heterogeneous landscapes, including alpine grassland systems, and again in subantarctic Marion Island, where environmental gradients create striking variability in soil properties, vegetation structure, as well as microclimatic conditions. So in these systems, even just small differences in elevation or aspect can lead to significant ecological differences. They're very, very different environments to work in, and there's a lot that you need to consider when designing studies to actually collect data here. Okay, so fine scale topographic complexity is a characteristic feature of these types of environments, and this has important effects on plants. So for example, in heterogeneous environments like these alpine grassland systems or in mountainous systems, we usually have steep environmental gradients over small spatial scales. A change in environmental conditions just over a small shift across a topographic gradient, for example, between north or south-facing aspects, may be equivalent to shifts in orders of magnitude larger than over altitudinal or latitudinal gradients. So moreover, at these fine spatial scales, you often have these unique landforms, as I mentioned earlier, and unique topographic combinations of soil characteristics that may comprise microrefugia that allow for the survival of isolated populations under current and future climatic conditions. So I did some field work in the grasslands that you see here from this picture. This is in the Drakensberg, specifically Golden Gate Highlands National Park. And I'm currently preparing a manuscript on this work for publication. But the point of this slide was just to show you that we have variation in landscapes, and this can happen over small spatial scales. So from this picture specifically, you can see we move from a very rocky area at the top with shallow soils, and over a small distance, we end up there where I'm standing at the bottom to an area where there's very deep soils. So conditions can change over very small spatial scales. And we have different soil properties in these different locations. It affects vegetation structure, as well as what I mentioned before, the different microclimatic conditions. And those are basically just conditions experienced by different organisms. So how do we go about collecting data in these highly heterogeneous or changing environments? How would we design a study to collect data in this type of environment? Well, there are several field protocols or techniques that you can use to survey vegetation in particular, and I've used some of these methods before in my research career. However, for this particular study in the Drakensberg that I'm showing you some results from, we used a fine-scale grid methodology to collect data. More specifically, we had 8 fine-scale sampling grids, and each of these grids, as you can see from the slide, were 20 by 8 meters long, comprising of 160 1-meter squared contiguous quadrants, and they were established and surveyed in different parts of Golden Gate Highlands National Park. The grids were located across the widest range of environmental conditions within a spatially limited but heterogeneous area. And each grid was orientated to sample maximal environmental variability, for example, covering a mesotopographic gradient or spanning a geological transition. So the grid in the picture covered a range of soil depths, rock cover, and steepness, again, which affect our conditions for our plants to grow in. And this sampling design, orientating the grids in this way to capture maximal heterogeneity, is a way to examine the influence of fine-scale heterogeneity without the effect of confounding factors like macroclimate or species pool. And within each of these grids, we had the 1-meter squared quadrants that we used. And just to give you a brief overview of what we did, we basically identified and estimated the cover for all vascular plant species, lichens and mosses. And we measured several abiotic variables. There, I give them in blue on the slide from each of those cells. And then from every second cell, because it's a trait-based paper that I'm looking at, but I'm not going to focus on that for the purpose of this study, we measured functional traits for the dominant vascular plant species in every second or third cell. And this ended up, we ended up having over 2,000 leaf samples that we collected per grid or per sampling location. And for each of these plants or dominant plant species, we recorded several trait values, which are listed there in black on the slide. We also collected soil samples from these grids, but unlike the trait samples and the species IDs, we can't collect soil samples from every single 1-meter squared cell because this is impractical. So soil samples in this case were collected from a subset of the quadrants, 18 evenly spaced quadrants per grid, to determine soil properties, things like texture, pH, carbon contents, cation exchange capacity, and other things. I then interpolated these data to the unsampled quadrants using bilinear interpolation. So you use those samples that you have to interpolate the data across your full grid. Then the last thing we did for these grids was we measured the rates of decomposition or carbon turnover, and we used something called the tea bag index, and it involved bearing rooibos and green tea in several cells in each of these grids. And why am I now telling you about ecology? Well, this is basically just to introduce you to heterogeneity before I move on to agricultural fields. This is just showing some of the results from the soil moisture analysis from these grids. There were 8 grids, if you can recall. And from the data that we've collected, I will now show you the 2 images. This is the first image. It basically shows variation in soil moisture within and between our 8 fine-scale sampling grids, with the darker colors, the blue colors, representing more soil moisture, and the lighter yellow colors representing drier conditions. So you can see at such a fine spatial scale, we still have quite a lot of variation within each of these grids, as well as between the grids. Then just as an example, I'll show you some of the results for soil carbon. Again, you can see lots of variation within grid, as well as between grids. So the point here is that we have lots of variation at fine spatial scales in these types of environments. And just like alpine grasslands or Marion Island, anywhere you go, I think that agricultural fields can also exhibit significant spatial heterogeneity. However, instead of natural gradients like elevation and aspect, perhaps agricultural variability could be driven by factors such as soil type, cropping history, irrigation patterns, and some other things. The fundamental challenges apply, and this is how do we design sampling strategies that accurately capture this variability. So if we recognize that a single sample in a heterogeneous grassland fails to represent the whole landscape, why should we expect 1 or 2 soil samples in a farmer's field to be any different? Understanding and managing heterogeneity is the core of good sampling. So whether we are studying natural ecosystems or managed agricultural fields, the goal is the same: to design a sampling strategy that captures this variability effectively while remaining practical in the field. So in this talk, I'll explore different sampling strategies that address challenges working in heterogeneous agricultural fields with a focus on sampling soils. I'll also discuss the methods to ensure representativeness and how do we determine appropriate sampling sizes, as well as manage things like spatial variability. And again, I can't cover much of the content from all of these topics, but I touch on some of the things that I have worked with and that I do know. So imagine 2 neighboring farms. The one has high crop yields and the other struggles with the poor soil health. What is causing this difference? Without proper sampling, we might never know. So collecting representative data is crucial for understanding soil properties, crop growth, and farm productivity. However, agricultural fields are not uniform. Sometimes they vary in soil properties, elevation, and management practices. And these factors can affect crop growth, soil health, and productivity. But we can only measure them accurately with representative sampling. This heterogeneity complicates soil health monitoring and the selection of representative samples. So our goal is to design sampling strategies that are statistically sound, practical in real-world conditions, and capable of capturing true variability within our fields. And the key objective of sampling is to ensure data representativeness. So we want to capture true variability in the field rather than getting misleading results from a few unrepresentative spots. We then want to improve reliability for decision making. So farmers and researchers use this data for soil fertility recommendations, precision agriculture, and sustainable management. And lastly, we would like to balance statistical rigor with practical feasibility. Sampling should be accurate but also efficient. So we must balance scientific precision with time, cost, and labor constraints. And a key question from field sampling is, how many samples do we need? And how many samples would be enough if I had to design a study? If we sample too little, we risk missing important variability. But if we sample too much, we waste time and resources. So the right balance depends on the field size, the crop or soil variability, practical constraints, and I guess also the research question involved. So generally, if a field is highly variable, we need more samples to capture that diversity. So more samples may be needed to accurately represent field conditions. A highly variable field may require stratified sampling to ensure that all zones are representative. And I'll discuss this stratified sampling a little later on. If a field is relatively uniform, fewer samples may be sufficient. We can also use preliminary surveys or remote sensing to adjust our sampling density. So the scale at which the scale at which sampling is conducted depends on the research objective, variability within the system, and logistical constraints. And that's from experience, from my experience in the field, those 3 things really do matter. So we need to balance our sample size with available resources. Sometimes it's better to take fewer well-distributed samples than to exhaust resources collecting excessive data points. So there's a trade-off. When we look at confidence intervals, for example, at 95%, It ensures that results are reliable and not due to chance, whereas statistical power determines how likely we are able to detect real differences in soil or crop parameters. So if we have more samples, we have greater confidence and power, but the trade-off is that there's also higher costs often and more time investment. So these are things that we need to think about when designing experiments or trials in the field. Existing studies have made efforts to obtain representative sampling or soil sample numbers to achieve accurate results. And these are just some of the approaches that I mentioned here, but there are others. The one is the coefficient of variation of soil properties that has been used to infer sample densities required within a field. Osborne et al. 2011 used the Monte Carlo simulation approach to quantify the number of samples required to understand bacterial community in pastures and revegetated forests. A similar approach has been used for forest soils where bootstrapping was used to simulate soil bulk density. And then we have something called power analysis that can be used, and it has been recommended and used to investigate the number of samples for properties like soil carbon and microbial biomass. So these are statistical approaches that we can apply to determine what a good sample size would be. Then besides the number of samples, another dilemma for soil health sampling or soil sampling in general in agricultural fields and other environments is where do I take my samples from? Soil sampling location, where should I be sampling? So if we look at agricultural fields, should we sample in row or between row? And it depends on how the field is structured. We may have different looking fields, so we need to think about where are we going to put our samples. A wide variety of soil microbial properties have been found to be greater in row compared to between row locations in different environments. And these results have been attributed to things like the plants in rows influencing the communities there. Soil pH was also found to increase near the roots of plants. So the plants themselves in agricultural fields will influence the patterns that you are seeing in the soil. As soil properties can change with sampling location, this may impact the number of samples required for accurate assessment. However, if the impact of sampling location on the number of samples is required, however, the impact of sampling location on the number of samples is required in research results is still unclear. So we don't know a lot about where would be the best location to place our plots and why we would place them there. I think it's a lot of going into the field and actually trying to look for yourselves to see what it looks like, to see if you can see different patterns, and then deciding, okay, which approach am I going to use? Where am I going to put these plots? So it requires knowledge about the system that you're working in. Then choosing the right sampling methods depends on the research question and the field conditions involved, as I just mentioned. So if we randomly sample, I list here on the slide different sampling approaches that you can take. So random sampling, regular sampling, systematic sampling, stratified sampling, subjective sampling, there's composite sampling, and adaptive sampling. If we sample randomly, we get an unbiased representation, but we might miss key patterns. In systematic sampling, such as collecting data along a grid or transect, we ensure more even coverage. With stratified sampling, we usually divide a field into distinct zones. For example, separating high and low yielded areas, and then targeting sampling in each of those areas, doing random sampling in each of those areas. Targeted sampling focuses on specific areas of interest, while cluster sampling helps when working with, for example, I would think multiple farms. The best strategy often combines multiple approaches. And adaptive sampling, the one I list there at the end, is basically where we adjust our sampling strategy based on what we observe in the field. And we can use tools such as remote sensing or GIS data to guide us as to where would be best to place our plots. So I haven't gone through each of these sampling strategies in detail here, but you can read about them. There's a lot written about them in the literature. Composite sampling is something that we often use for soil analyses. So that's basically when we combine samples from different locations to form one representative sample for that area. So these are just the different types of ways that you can position plots and the different sampling methods that you can use. Here I just show a few of these that are commonly used. The first one shows random sampling. So every location, if those blue dots represent locations, every location has an equal chance of being selected. In stratified sampling, which is showing in the second diagram there with the red and blue dots, the field is divided into zones based on known differences, for example, soil type, elevation, or past yields. And then from each of those zones, sites are randomly selected for sampling. GIS might enable you to analyze and visualize spatial data, which can help dividing the fields into homogeneous zones based on factors like soil type, elevation, land use, or past yields. So use these tools. You can create accurate maps of the study area, identify strata, and select sampling locations more effectively. Then there at the bottom, I just have some text for stratified sampling. You can basically use an aerial image in GIS. You can print it out and you can overlay a transparency sheet with random points on that sheet over the photograph of the study area. You then align two sides of this transparency overlay with the photo and mark the points on your photo with a pin. You then move the transparency to align with two different sides of your image and mark the new points. And there are 16 possible ways to align the overlay. And at the end, you repeat the process until each area has at least one sampling point in it. So that's a nice way to make sure that you capture variation that may otherwise be uncaptured through just normal random sampling. And here I've just listed the different sampling types with both their advantages and disadvantages. So there are many different approaches to collecting representative samples in a research plot. And I've gone through a few of them for you. I'm not going to go into this slide in detail because I don't want to use up all of my time. I see I'm running short on time. Gerard, just stop me if I go over.
No, you're fine, so thanks, Morgan. Appreciate it.
Okay, cool. And there's some other protocols as well. So those are not the only ones that I mentioned before. There are a few others. I've just mentioned them here briefly on the slide. Clustered sampling is a sampling technique where the entire population is divided into smaller groups known as clusters, and a random sample of these clusters is selected for study. So within each of those chosen clusters, either all of the members or a random sample of the members is surveyed. And this method is useful when it's difficult or costly to conduct a survey across an entire population, but the population can be divided into smaller groups that are more accessible or easier to study. Then grid sampling, I showed you an example from the study that I used, the one that I'm busy working on for publication now. That's an example of grid-based sampling. Then we also have something called transect sampling, and this is when samples are taken along a linear path or transect across the study area, and it's often used in ecological or environmental studies. So when studying environmental gradients, this is quite a useful approach to use, obviously, because you can go along in a line following an altitudinal gradient or a gradient in soil properties or elevation or moisture. So yes, it is a useful technique. And if you require any more detail about any of these methods, you can definitely contact me. I'm happy to give you background information on them. I obviously couldn't share all of it with you now because of time constraints. But yes, I do have more information if you would like that information. The next thing I'm going to talk about is managing spatial variability and confounding factors in the field. So this is an essential step in ensuring the accuracy and reliability of field estimates or studies. Spatial heterogeneity is a major challenge, as variations in factors like soil properties and environmental conditions can lead to misleading results if not properly accounted for. So techniques such as geostatistics and spatial interpolation or rigging help estimate and model these variations, providing valuable insights into spatial patterns across the field. Additionally, you can use things like drones and satellite imagery, which allow researchers to capture these variations before fieldwork actually begins. So I think it's important to know your study site before you actually get there. So oftentimes when I do fieldwork, I go and visit the site first to see what I'm working with. And from there, I start to think about my experimental design. This can help guide your sampling strategy and improve the efficiency of your sampling. Transect sampling is another useful method, as I just mentioned. It captures gradual changes across a field, ensuring that variability is properly represented. At the same time, it's crucial to account for confounding factors like environmental influences, rainfall and temperature. You could consider things like management practices, fertilization, irrigation. All of these factors can maybe mask or exaggerate the effect of other variables in your models. So it's important to know how to deal with these confounding factors when you are modeling your data to try and answer a specific question. So employing strategies like paired comparisons or spatial controls can help isolate the effects of these confounding factors and ensure that observed changes are attributed to the right causes or variables and not external influences. And there are some ways that you can sort of try and better manage spatial variability or confounding factors that I didn't mention on the previous slide. But this is more of when you get to the stage where you want to analyze your data, you can consider things like this. First is multi-scale analysis. So why is this important? This approach helps identify spatial trends that may be overlooked at a single spatial scale. So enabling a more comprehensive understanding of spatial variability and confounding factors. So basically with this approach, you incorporate data or you collect data at multiple spatial scales, local, regional, and landscape levels. But just remember, you need to keep your key objective in mind. So don't do this if it's not a key objective. But this could provide a better understanding of spatial patterns. Small-scale studies might focus specifically on localized variability, whereas larger scales capture broader landscape-level patterns. And then we have mixed effects models, which I love to work with. So these models, I had to use them for nearly every single project I've ever worked on. These models can be used to account for the impact of unobserved spatial factors that vary across study areas. This can be especially useful when dealing with large-scale data sets where it's difficult to account for every spatial factor directly. So they provide a statistical way to control for variability that may arise from unmeasured or unaccounted factors. Very, very useful tool to have if you are working in heterogeneous environments. And then lastly, we could consider temporal analyses. So accounting for temporal variability or changes through time alongside spatial variability can help control for factors such as seasonal variations. And if we think about agricultural systems, maybe crop growth stages and crop performance through time. So over time, environmental conditions and management practices can affect soil and crop health. By considering temporal changes into your models or doing maybe repeat sampling in time, you can separate out the influences of temporal variability from spatial variability. Okay, so a great sampling plan can fail in field if the execution is poor. Pre-sampling field visits can help refine the sampling strategies. I would suggest if you want to sample, go and walk in the field beforehand and visualize or assess variations in the landscape that you can see. Maybe you can see differences in soil visually. Look at the vegetation. Does the vegetation change in different areas? And other factors that might influence the study. So just go have a look. It's the best way to see what you're working with. And this helps you identify areas of high variability and adjust your sampling plan accordingly, ensuring that your data reflects the true spatial heterogeneity of your environment. Again, as I mentioned earlier, we use GIS tools to assist us with these types of designs. GPS tracking specifically ensures consistency when collecting multiple samples. So make sure that you have a deep GPS unit to record where you are sampling from. Accurate georeferencing of sampling locations with the GPS ensures that the spatial distribution of the samples is well documented and can be precisely mapped. So if you want to overlay it onto an image, you can maybe see, okay, does this actually look good? Am I capturing enough variation? Additionally, working with, for example, in an agricultural setting, if you work with the farmers, I think that would be invaluable as they know their land best. So their insights on historical yields, soil conditions, and problems in the area can improve your sampling accuracy. And finally, we need clear communication with the people that we're working with. If you're working on farmlands, make sure that your communication with the farmers is clear so that everyone knows what's going on, and so that we know that everyone gets a mutual benefit from the research. And that's it. If there's any questions, I'm happy to take them.
Thanks, Morgan. That was really, really great. A lot of very valuable information and actually a lot to digest.
Yeah.
So, it's striking to me how much there is to learn about sampling design. And it's probably why, at least from my perspective, and I think you've just proven that, is that it's actually really important to have experts involved in your projects, especially when it comes to thinking about how sampling should be done. So I have a few key takeaways, but I first want to open the floor for general comments and questions. And we do have a few minutes before we continue with the next presentation. So if there's anybody from the participants who are keen to make a comment or ask a question, please go ahead. You can just raise your hand and then I'll give you the mic. Anae, hi Anae, please go ahead.
Hi, everyone. Thank you, Morgan. It was such an insightful talk. Maybe if I just can get a comment from you, working with something like herbicide residues in soils. In the Western Cape, we have giant agricultural fields, and sometimes we have to test for residues. And obviously, products sometimes move into the soils to different depths. And do you have any comment? Have you maybe worked on something like that? Obviously, you have taken in everything that you've said, but yeah, any general comment from your side?
Hi, Anae. Thanks for your question. I will say that's quite a tricky one for me, as I've never actually worked in agricultural systems, so it was a new one for me. But I suppose the issue with the chemicals being absorbed into the soil, you could do your general soil analyses, but you would need to account for the effect of those chemicals in your statistical modeling, I think. So you would need some sort of way to account for that being a confounding factor in your analysis.
Perfect.
And yeah, if I
Thank
can find any sort of information on it, I have come across a few papers which I haven't read yet. I can maybe forward those on to you.
Thank you. I would appreciate that because I sampled like the top layer and then a little bit deeper, and then I could already see a distinction. So I was just wondering if you have any info. So that would really help. Thank you.
Does it influence the actual soil properties?
I doubt it. I don't think so. No.
Then it should be okay. Then it shouldn't be an
Yes.
issue. If it's not influencing what you're measuring, it should be fine.
Perfect. Thank you.
Thanks, Anne. So anybody else who'd like to ask a question, make a comment? So I'd like to share the few, I've got 3 key takeaways, and I think, I mean, there's a lot to learn from this presentation, but 3 points I think is quite important. You made a statement, Morgan, balancing statistical rigor with practical feasibility. I think that's absolutely key, right? It's not easy, I think, because you have to weigh many things. Logistical challenges, also the cost of taking an X number of replicates and analyzing those samples. But then also, you know, the more replicates you have, the more replicates you have within a sensible design, the more statistical power you have in the end.
Yes, I can use an example, specifically a challenge that we faced with some of the Drakensberg work that I've done in Lesotho. Actually, we sampled there, and just collecting soil samples, they're so heavy because you fill those canvas bags with the soil samples. And if you had to collect for every single grid, it's 160 bags of soil that you have to carry down from your site. It's about a half an hour hike to the car. Just getting the soil samples down is a mission, and then getting them through the border posts is impossible. So in that event, you can't sample extensively. You have to use other tools, interpolation tools, to try and use that data to get an idea of what the values would be across an entire grid. So in that case, it's not feasible because you just can't do it. There's no funding to process all of that data.
Yeah, absolutely. And I think in the agricultural landscapes, I mean, sometimes, especially if it's later in the growing season, you can't enter the field with a vehicle, so you have to walk. And if you're working on very big fields, it becomes very difficult to travel across the entire field to sample. So I think there are similar challenges also in agricultural settings. So maybe linking to that, another key takeaway that I think is very valuable is that it's important to understand the landscape you're working in. So, I mean, you kind of need to define what it is that you need to know about the landscape. But if we're using agriculture, I mean, you really need to understand what's the potential of having a very homogeneous versus a very heterogeneous environment. And for example, soil types or major differences in soil textures. So I think that's another key point when undertaking the study, is to really understand the environment that you'll be working in. Maybe a final key takeaway is that your sampling strategies should really link to your research question. I mean, you mentioned inter versus intra-row sampling and the potential effect that might have on your results. So, I mean, and that goes back to the question that you're trying to answer. And I just think it's very important to always have that question in mind when you're actually working on your sampling design.
Yes. And even though I've never worked in an agricultural system, I can see that as a potential factor that has to be considered here, just by the layout of crops here. Because plants in themselves have the ability to modify conditions around them. Whereas if you look away from the plants, the conditions might be completely different.
Yeah, and I think there's a general idea or perception that agricultural landscapes are homogeneous, but they're not. I mean, we know from work we've done is they can be highly variable, and just something like soil type or soil texture, that really has a massive influence on the biology in the soil, and also how the plants respond to that. So we know that agricultural landscapes, although they appear homogeneous, they're not necessarily homogeneous, especially the soils. Maybe just one question before we continue with the next presenter. You know, you mentioned determining sampling sizes before a sampling event, but I mean, you do need preliminary data to do that. And that's one of the things I've always had a hard time doing, is how do we actually, I mean, what is an effective way of generating the necessary data to actually determine your sampling sizes using statistical approaches? I mean, do you have any additional thoughts on that maybe?
I've never actually used the statistical approaches to quantify my sample sizes beforehand. What I tend to lean on is more looking at other studies that have tested similar questions, looking at their sample size, whether there were any issues with their statistics because of their sample size, and adjusting my approach accordingly. So you basically want the number of samples that you collect to be representative enough to answer your research question. And you could employ one of those statistical techniques beforehand, but I think it's probably better to go into the field first, look what you're working with, is it feasible, and then have a look at what other people have done, either on a similar topic or a topic that's sort of parallels yours. See how many samples they collected, because you know that their work has obviously undergone the rigors of peer review. So if there were any issues, it would have been highlighted. Then based on their findings, their results, you can see, would it be better maybe to include more samples, or is it okay to use the same amount that they used? Or do I want to use a different amount entirely and then justify that using a statistical approach?
Yeah, I think that's feasible. And I agree with that, because sometimes just going out to the field and doing preliminary sampling just to determine effective sizes, basically, that would be challenging, I think, in many cases. And I actually don't recall really seeing a lot of papers that followed this approach. I think it would be great if you were able to do it, but I just think from a practical point of view. So I think the approach that you mentioned is a good compromise to that.
Yes, and it's more like playing it by ear when you go into the field. Because oftentimes I've had events on Marion Island where you go into the field expecting to collect data in a certain way. I can give an example. I took a vacuum cleaner into the field with me to vacuum cushion plants to see how they trap seeds. And you go into the field with this idea that you're going to do this many samples today, and then something happens, and then you have to change your plan completely. And in my case, the vacuum cleaner battery life lasted only 10 minutes. So I had to go out into the field, walk 10 kilometers to my study site, vacuum for 10 minutes, walk back, charge my vacuum cleaner, walk out for 10 minutes, do my sampling, and come back. And then eventually I had to adapt and get an engineer to put a car battery onto the vacuum cleaner and then proceed with the sampling. So you have to adapt and change as you go.
Yeah, I agree. And that comes with experience, right? So, and maybe that links up to one of the very first comments I made after your presentation, is that we need experts involved. So, I think in my mind, you either have two options. You become an expert yourself in stats in the way that you do your sampling designs, or you surround yourself with people that are well knowledge in this field, and you kind of have them on speed dial if they're not in the field with you. That's at least from my point of view, two options that are feasible, that could work.
Another option that you could use, which I have tried in the past as well, is you could design a study and say you're going to do this many samples here, this many samples here. Then you generate a dummy data set for yourself. You then use that dummy data set to run some preliminary analyses and see if it gives you any problems.
Okay. Yeah, that also sounds an interesting approach. So Morgan, I think thanks a lot again. It was a lot of very valuable information. And if it's okay with you, I'll request the slides from you, maybe as a PDF format. And I'll share that along with the recording of this meeting with NSSA members. And I think we'll probably
everything. I couldn't cover all the methods in details, but if anyone wants more information, I'm more than happy to send that through to you.
Yeah, no, I appreciate it. Thanks a lot, Morgan. Yeah, anything you want to send through, you're more than welcome. We'll share it with all the NSSA members.
Okay. Thank you. Thanks for the opportunity to present.
Thanks, Morgan.
Thank you.
So with that, let's move on to our next presenter. So Liesl, we're very excited to hear what you have to share with us. So if you can maybe just test and see if your sharing option is working.
It says couldn't share your content.
Okay.
Let me try again. You see it now?
Okay, that works. I can see your title page and it's the full page. I think it's working.
Okay.
Yes.
I can't see the chat or anything now, but if anyone has a question, you can just stop me anywhere. Thank you very much for inviting me. It's a privilege to be able to present to you today. I hope it will be relevant for everyone. So I was asked to give some background on experimental design and its implementation in field and greenhouse trials. So it's a bit different from what Morgan presented on. So, sorry, now it doesn't want to scroll. Okay. So the first thing that we must understand is why we need experimental design. So in our case, we're going to apply something to either our field or our plants. So we want to see what is the effect of that, and is that effect significant? So if you compare the effects of different treatments, you need to know if those effects that you observe in the crop or in the field are truly a result of the new treatment that you have implemented and not simply a product of the natural variation that occurs in every ecological system. And Morgan has shown you lots of pictures of that natural variation that we need to account for. So experimental design is the process of carrying out research in an objective and controlled fashion. So we try to control as many factors that we can so that precision is maximized and specific conclusions can be drawn regarding a hypothesis statement. So whatever you decide you want to test. So to achieve this, we have two very important components that are essential. And the first is replication, and the second one is randomization. We cannot do an experiment without either of those. So replication is needed so that we can estimate the error variation of the experiment. So we need sufficient number of degrees of freedom for error, which we will go into later again when we go into specific examples, but we generally recommend 16 to 25 degrees of freedom for error. So from this, you will be able to get a reliable estimate of treatment means. So there are a few basic experimental designs that I'm going to discuss today. The first one is the completely randomized design. It's also known as a simple random or a one-way design. In this experimental design, you make no attempt to allow for position. So the treatments are randomly assigned to your experimental units, which can be plots in a field, or it can be pots, can be seedling trays, whatever you decide on for your experimental unit. So here I have an example of a completely randomized design where we have 5 treatments, and each one of those treatments are replicated 6 times. So 5 times 6 is 30. So in total, we have 30 experimental units. And there I've just randomly allocated the treatments, which are my colors, to my plants in the pots. So you can see it's completely random. In the bottom row, I have 1 red plant. In the top row, I have 3 red plants. So it's just a completely randomized design. You need to know how to calculate those degrees of freedom for error that I mentioned previously to see if your trial is large enough. So this is just a skeleton analysis of variance. I don't have all the columns in for simplicity. So for treatments, degrees of freedom is always your number of treatments minus 1. In this case, we had 5 treatments, so the degrees of freedom is 4. In total, we had 30 pots. So 30 minus 1 is 29, and then your error will be the difference, which is 25. So if you decide to do this trial, it will be large enough, but we usually don't recommend it because usually there is some source of variation that you have to account for, which brings us to a randomized complete block design. The abbreviation you can see there in brackets. People get confused if they make it R, B, C, D, and I've seen lots of variations. So complete block just means that all your treatments are randomized within every block. So incomplete block would be that you have some treatments in one block and some in another. So the complete tends to confuse people. So it just means all the treatments are in all the blocks. So the simplest example of the use of local control is the randomized complete block design. So here you divide your experimental area, whether it's the greenhouse or the field, into a few blocks. The number of blocks are equal to your number of replications. And then within each one of these blocks, you will randomize your treatments or treatment combinations if you have a factorial experiment. So, as I've mentioned, a block may be an area of the field or greenhouse, can be a table, a part of a table, it can be a shelf in an incubator, and it can also be time. So I'm going to spend some time on blocking, because one mustn't just block because someone told you you must do a randomized block design. You must block in a specific manner to try and take gradients into account. So in field trials, there could be soil fertility, soil moisture, soil type, a slope that you have to take into account. In a glasshouse trial, it could be cold or warm air from the air conditioner. You can get shade in the morning or shade in the afternoon. In a laboratory trial, it could be the time of day or operator changing. So you would want one person to do the analysis on a block or a replicate. You won't want another person to come in halfway and analyze those samples. So similarly, if you harvest large plots in a field, and you can't do a whole trial in a day, then you would harvest whole blocks at a time. By doing this, time effects may be attributed to blocks, and it will not be confounded with your treatments. So the purpose of blocking is to compare treatments within groups of experimental material which are as homogeneous as possible. So if you want to do a trial on trees, you might receive 30 trees from a nursery, and there might be a lot of variation in the size of the trees. So you don't want it to by accident happen that you apply treatment 1 to all the large trees and treatment 10 to all the small trees. Now you think the treatment is great, but it's not that. It was because of the size of the trees. So what we usually recommend is that you arrange your trees from the largest to the smallest tree, or the other way around. And then you will subdivide your trees into the number that you need for a block. So the smallest number of the small trees. So you have 10 treatments, you take the 10 smallest trees and you will randomly allocate your treatments to the 10 smallest trees, and you'll do that for all the groups until you get to the largest trees. That way each treatment is assigned to a small tree, a medium tree, etc., up to a large tree. The same goes for animals, which I don't think you really work with, but you can also sort them according to their body mass or lactation stage. So a specific weight or stage will be assigned to a block. If you work on bulbs, you should also sort the bulbs according to size, and then block according to that size, so that size is not confounded with your treatment. So the real aim is that there must be as little variation as possible within a block. So if you do a trial where there is a gradient, like a slope, then if the direction of the slope is downwards, you would not lay out your blocks like this. You must lay your blocks out with the slope direction so that you have a block at the top of the slope and your last block at the bottom of the slope. The same will be if you arrange treatments in shelves on a growth chamber. You want all your treatments on every shelf so that you take into account that difference in temperature or light that you might get between the different shelves. Sometimes you have more than one pot or plant per experimental unit. In this case, I have 10 pots or plants. So if you have a temperature gradient going that way, we've arranged our blocks to go from left to right. So if you arrange your treatments like this, treatment 1 will be closest to the cooling fan and treatment 6 will be farthest, even if that it is in block 1. So this is not what you want. What you would do is you would arrange your treatments so that treatment 1 has a pot closest to the fan and furthest from the fan in block number 1, and we carry on like that in the other blocks. So that is the correct way. So if we look at the skeleton analysis of variance for a randomized complete block design, you'll see we now have an extra factor for a source of variation, which would be the block. And the block degrees of freedom is calculated as a number of blocks minus 1. So because we add another factor, you will see that your error degrees of freedom will be less than what you would have with a completely randomized design, because in a completely randomized design, the 3 would have formed part of your error, so it would have been 12 degrees of freedom for error. So you can either increase the number of replicates or you can increase the number of treatments if you want to increase the number of degrees of freedom for error. So if we still keep it at 4 blocks, but now add another 4 treatments, now we have 8. You will see you have 21 degrees of freedom for error. And if you then still increase the number of blocks, you will be able to have 35 degrees of freedom for error. So there are different ways to increase the size. You don't always have to do more replicas. You can just do more treatments. So if you expect a lot of variation in your response measurements, then you would maybe have to increase the degrees of freedom for error to say 20 or 25 degrees. And then you can see from this that you have to know how to calculate the degrees of freedom for error for the experimental design that you use. The third type of experimental design is the Latin square design. This design is used to control local variation in 2 directions, as opposed to a randomized complete block design that only controls variation in 1 direction. So here on the left, I'm going to show you the randomized complete block design. We have 5 treatments randomized within 5 blocks. I've laid the blocks out in the direction of the variation. If we now rearrange this randomized complete block design so that each treatment occurs once in a row and once in a column, and you can verify that, you can see yellow, all our treatments are in this row and all our treatments are in the first column. So then we say we can control variation in 2 directions. What will happen is that in the randomized complete block design is that you can get the same treatment below each other, which would not happen with a Latin square design. So the restriction upon the use of a Latin square is that your rows, columns, and treatments must all be equal in number. So that makes it a bit difficult. And the smallest design that is really feasible is a 5 by 5, but that only gives you 12 degrees of freedom for error. So if you use a smaller square, like a 4x4, you will have to repeat the squares to get enough degrees of freedom for error. So you must take into account whether the increase in labor and materials is worth it, because it can become very costly. So because the Latin square eliminates effects of fertility or positional differences in two directions, it's often preferred by entomologists who cannot guess from which direction an insect infestation might come. So they often use it in orchards. So they would lay out more than one Latin square, and then it doesn't matter from which direction the insects come, the design will be able to account for the direction of infestation. So here's another example of a Latin square design. So Latin square designs are available in books, or these days we can Google them and tell Google, I want examples of 6x6 standard Latin squares. So the important thing to remember is that you cannot use it exactly like you have it there. It must first be randomized. And the way we randomize it is you randomize the columns first. So here you can see, for example, the third column has moved to the front here. And then after you randomize the columns, you also randomize the rows. So how are we going to apply the 6x6 Latin square? Is we are going to test the effects of 6 temperatures on the toxicity of an insecticide. And because we only have 6 growth chambers available, we can only test a temperature at a time. So here we're going to have a chamber, and then we're going a chamber for each temperature, and then we're going to randomize which chamber is set at which temperature every week. So in the end, every temperature would have been set at each one of those 6 temperatures. So after you've randomized your rows and columns, you're going to decide, am I going to use the rows as my weeks and the columns as my chamber, or the other way around. So a chamber would be set at a specific temperature. So you'll randomly decide which letter is which temperature. So in week 1, chamber 1 will be set at temperature E, which might be 30 degrees. And then in the next week, each one of those growth chambers will be set at a different temperature. So if we look at a comparison of the area degrees of freedom for the 3 designs that we've already looked at, you can see as you compensate for a source of variation, the error degrees of freedom decline, because each time you added another factor to the analysis of variance. So for 4 treatments randomized 4 times, you can see for a completely randomized design, you will have 12 degrees of freedom. For randomized complete block design, you will have 9. And for a Latin square, you will only have 6. For a 5x5, you end up with 12 for the Latin square. And for a 6x6, you have 20 degrees of freedom for error. So this is what an analysis of variance for a Latin square would look like. You will have a row, source of variation, a column, and a treatment. And you can see they will always be equal, because we said row, columns, and treatments must be equal. So this is for a 4x4 Latin square, a 5x5, and a 6. That's why you see a 5x5 Latin square is actually still small. Now, lots of times you have more than one treatment that you want to compare at the same time. You want to combine different kinds of treatments, and this is now known as a factorial treatment design. So where the effect of more than one factor is studied, it's known as a factorial experiment. So here we have an example of a trial layout of a randomized block design, but our treatments are going to be a factorial. So here the researcher wants to test the effect of 3 rhizobia treatments and 2 water regimes. So 3 times 2 is 6. So we have 6 treatments combinations that we are going to randomly allocate to 4 blocks. So here you can see our 6 treatment combinations randomized within our 4 blocks. So it can firstly be seen as a randomized complete block design consisting of 6 treatment combinations. And then the ANOVA will look like this. We have our 4 blocks, which will give us 3 degrees of freedom for block. We have 6 treatments, and our error is 15. Sorry, that was the wrong one. So what happens now in the factorial analysis of variance is we break up the treatment, which has 5 degrees of freedom, into the rhizobia, the water, and the rhizobia by water interaction. You can see the degrees of freedom is 2 plus 1 plus 2. It's 5, which is exactly equal to what we had for our treatment. So we first test the main effects and then the interaction. And then the last design that I want to show you is a split plot design. So a split plot must only be used if it's really necessary, and you will see why if I show you the analysis of variance table on the next page. So if we take that exact example that we just had, but now we say it's difficult to randomize the water treatment. So we make water our main plot, and then you can see each block has now been subdivided into 2. We randomly allocate the water treatment to the 1 half. So here we have water treatment 1, and within that we randomize the 3 rhizobia. So that is done for all 4 the blocks. So now we have 2 main plots, which is our water regimes, and within each one of those whole plots, we have our 3 subplots, which is the 3 rhizobia. So because rhizobia are randomized within water and water is randomized within a block, we now have 2 error terms in the analysis of variance. So I've highlighted it in red. So you can see usually with a split plot, the degrees of freedom for the first error term for the main plot is usually quite small. It doesn't have such a big effect on the subplot factor and the main and subplot factor interaction. So if we want to be able to have more degrees of freedom to test water main effect, you can either add more replicates or you can add another main plot treatment. So you can see if we just add 2 more blocks, it only increases from 3 to 5 for the first error term. But if we add another treatment, it actually doubles. So, sorry, I hear someone. So then I just wanted to highlight the difference between replication and subsamples, which is very important. Our replications must be independent from one another. So here I have an example of a pot trial in a greenhouse. The photo was taken 10 days after the seeds were sown. Pots were labeled, and we have different barley genotypes sown in these pots. After germination, the seedlings were reduced to 7 plants per pot. Now, in this case, a pot is an experimental unit, And seedlings per pot are some subsamples because they're not independent. So treatment should be randomized. So you can't have more than one pot of a treatment that you keep together unless those pots that you keep together make your experimental unit. So if you keep 3 pots of a treatment together, then those 3 pots are subsamples, and the measurements on the 3 pots will give you a representative value for one replicate of that specific treatment. So somewhere else, you will have another 3 pots of that same treatment, which will be your second replicate. And that's it from our side. Okay, thank you very much.
Thanks, Diesel. That was also very, very informative and I think very useful for a lot of the participants in this forum meeting. So I want to open the floor. So if anyone as part of the participants list have a question, you can just raise your hand. And I have Mika and Milat in my office also asking questions, but let's check for the participants list first. Anyone raising their hands? Oh, okay, so Mika, if you want to go first. That's fine.
Can I say hi, Liesel?
Hi, Mika.
Liesel, I want to ask a question. So we had a trial we planted maize. Now it's a nematode trial. So in fact, we should actually take samples before the time and then allocate plots to do that. But we do each plot actually the treatments, but we don't do that. So we plant the maize and then we hope that the nematodes are fairly evenly distributed. What I had in that field, it's a small field, it's in Nelspruit, is that one strip in the field had much larger maize plants than the others, but it went through the reps. So it was a completely randomized block design, but I have one area where everything is much taller than the rest. How do I deal with that? Can you use a factor to calculate that in, because it disturbs the yield and probably also nematodes because there is a bigger root system. So in theory, you can have more nematodes there.
The only thing that I think you can do after the fact is maybe use the size of the maize plant as a covariate so that, you know, when you analyze the nematode data, that maize plant size then compensate, like you say, because a bigger plant will have a bigger root system, so you would expect more nematodes.
Okay, and how? So you said block design, you could a block design have been a better choice?
I think if you had known before the time that piece
space
that yes would have a higher yield, then you would have laid your one block on that higher yielding plot so that you have all your treatments in there. So it would have helped if you had known before the time that it was more fertile soil. You know, there might have been something in the soil before the time, you know, maybe because of a previous trial that had been there that would have caused that. So I don't know if it would have been possible to sample the soil before the time to, you know, maybe that you would have known that you would expect plants to grow better there.
So it's really quite small, and no, I had not seen this before. So when we planted tomatoes or potatoes, we did not see this huge effect. It was only in the maize that we suddenly saw, but the maize is a quarter taller than the rest of the plants, and we had several different treatments, but not all the treatments in that area. So I don't know how to compensate for it because I don't even know what to measure what it was in the soil. So I don't know whether we would have picked it up, but we would have to do a soil analysis, nutrient analysis, which we don't do for nematode trials.
No, and it's very expensive, I know. So,
Yeah.
but did you measure the maize plants as well?
I don't think so. We did yield. We did collect yield, and it was more in that region. So the main thing is still nematodes, and because I think it was only over 1 rep, I just used with the averages. But I did feel that something here is not completely fair. I mean, but we couldn't do it. So the other option is to take out 1 rep if you have enough reps, or is that not possible?
Yes, but you said it wasn't a whole rep.
Yeah,
that
it
was
wasn't the
free.
whole, it was true. So because the other option would be to add a few more reps, but only use the most representative ones. Because it's a small plot and you still have a variation in the soil.
Well, if you know about that beforehand, yes.
Yes.
So you can't do that after the fact.
No, you just do more and then you use the sixth most.
Is that allowed, Liesel? It's a good question, I think. I mean, from an experimental side.
Only if it's a huge outlier. Yeah. So usually when we analyze the data, we would ask, is this value correct? Because it's so far out from the rest, then one would exclude it. But you know, maybe you never know when you analyze that data, maybe because most of those treatments did fall in one of the block effect might remove it. You don't know. So, the analysis sometimes does compensate. So, but I don't know if we've analyzed that data already, Mika. Have we?
Yeah, it's old. It's
Oh,
a
okay.
long time ago, so I can't even remember what were the results.
Yeah, but what should help then is if you could take the maize yield and use it as a covariate, for the nematode data, then it would take into account that some plants were larger than others. So
Liesel, just out of curiosity, I know there's an analysis, I guess, based on the ANOVA approach that compensates for covariates. What's that called again?
it's analysis of covariance. So you just add that variable as a linear factor in the analysis of variance. And
So
then,
is that an ANCOVA?
Yes.
Okay, it's good. Now I'm noting that down. Okay, Milad, you had a question.
Thank you, Lisa, for a very informative talk. Actually, my question is about the degree of freedom for error. So I want to ask, sorry, my information regarding the study is quite limited. What is the fair or a strong number for degree of freedom of error? So when we do the analysis, how we know that the selection of the design and the analysis is suitable for our project based on the degree of freedom for error?
20.
Okay, yes. Sorry, I only had 20 minutes, so I didn't want to put too much information in. But if you do analysis of variance, you do that F-test. So based on that, if your degrees of freedom for error is too small, you need a huge difference between means to be significant. That is why, from experience and literature, we know that if you have at least 16 degrees of freedom for error, your analysis should be able to pick up significant differences between means if they are there. Because if you do an analysis of variance and you only have 4 degrees of freedom for error, then your treatments will have to be very different from one another for it to be significant. I don't know if I've answered your question.
Yeah, so basically what I understood, the minimum should be 16 to get the statistical differences.
Yes,
Yes.
then you could feel confident that your trial is large enough to pick up differences if they are there.
Okay.
5 times.
Liesel, if I can ask the opposite question, what if you've got so many replicates or treatments that your degrees of freedom is too big, and you would, because we've had this discussion before, that you can even create a situation where you have too many degrees of freedom, and you might even potentially pick up differences as significant, but the effect is not that important. Maybe can you just comment on that, maybe on effect sizes?
Yeah, that's why when you do a factorial with lots of factors, it always makes sense to look at. This is now another part of the analysis of variance table that I didn't actually show, but your sums of squares as a percent for that specific factor as a percentage of the total sums of squares. So usually if it's less than, you can decide 5 or 10%, it doesn't actually make a difference. But what also happens if you have a lot, so many replicates, then sometimes you have a lot of variation in your data and you don't actually get that many significant differences. So it depends on the type of data that you collect. I know in soil data, there is usually quite a lot of variation. So I would recommend that you look at the percentage sums of squares of the total sums of squares, and just see, even though this is significant, the nitrogen accounted for 80% of the variation. This one is only 5%. So let me focus on, for example, the nitrogen.
Okay, so that makes sense.
So what is the considered as a very high degree of freedom for error?
No, we don't ever mind if it's too large.
It's a good thing.
It's too low, yeah.
You know what happens. I've never had a problem with that. The only problem, if I get data and I analyze it and everything is significant, everything, you put in all the variables and everything is less than 0.001, then you can go back to the researcher and you can ask, and you always pick up that it's subsamples, that they maybe took a soil sample, analyzed it 3 times, and that's your 3 reps. So I've never had it that you had true replications that were too many. So, one must always make sure just that your replications are independent. We've also had an example of where they do a nice field trial and they take soil samples and then they make this 5 reps, 1 rep for the soil analysis, do a composite sample, and then you understand that's where the problem comes in. So replicates are supposed to be independent, and I've never seen too many replications in any data that caused a problem.
But what about too many treatments?
It just makes it difficult to interpret, I think. Yes, especially if you combine a lot of factors and you get a lot of significant interactions, then it just makes the interpretation difficult.
Liesel, thanks. I really appreciate that. I think we all appreciate the insightful info. I want to just list a few key takeaways again, and then I have 2 quick questions as well, and then I'll again open the floor for any final questions. So I think as we saw also with the previous presentation, it's really important to understand the landscape that you're working in. So in this case, especially any gradients that there might be in the field or the greenhouse. It's also important to consider the statistical power of your experimental design, looking at the degrees of freedom and potential ways of increasing the degrees of freedom if necessary. And then the final point, which again was also true for the first presentation, is that I think and we tend to do this after the fact, but it's really important to have a biometrician or statistician involved from the beginning of your project, rather than giving someone the data after you've analyzed the results and then trying to figure out what's going on and how to make sense of it. So including an expert from the beginning. If you're not an expert yourself, like myself, that's probably something to consider. Liesel, just 2 quick questions. So we spoke about variability when there's a gradient, but what about a field where there's high variability, but not in a gradient. So it's just completely random. I'm thinking about soil types, for example. I'm assuming the CRD design is the one you should choose then, but does that always work, or is there something else we should consider as well? Or should you just choose a different field?
Could it be possible to block on soil type? That you do all your treatments on a specific soil type within a block.
Okay. So if you're doing a block design, you could have one block on one soil type and then another block on another soil type.
Yes. Yes.
And it doesn't matter if they're spatially separated. They're not
No,
aligned in any way. They're spatially separated. That's okay.
that's okay. Yes.
Okay. That's a good
Then
point.
yes, then the block should remove that effect of the soil type. Yes.
Then you only have 2 recs.
Well, I mean, if you have 5 soil types, then you have 5. But I guess that's a good question that Mika's asking. Can you do 2 blocks on one soil type and 3 blocks on another? Would that not cause problems?
Yes, no, that is just as long as the soil type within a block is
Oh, okay.
as homogeneous as possible. So
Okay,
that,
so
yes,
accounted for basically.
yes.
And then the last question, something we've had to deal with students especially before. So how do we ensure unbiased randomization? Because sometimes we look at our randomized design and we're like, well, we really want to move this one block somewhere else or this one plot somewhere else. So what do you recommend? How is there software we can use, or just a simple approach in Excel? From your experience, what's the best way?
Yes, there is well, there is software available, or just ask someone else to do the randomization for you, because I mean, they don't know, they are unbiased. So what is best is if you ask a biometrician to do the layout for you, because then, you just say, I want to do it in a glass house. Then I would ask, what is the layout, where's the tables, all of that, and then try to fit everything in. But I'm sure there should be software available that is free for anyone to use to do a randomization. But if
Okay.
someone would ask me, it doesn't take me long. I would love to do it. I'm always available.
Okay, that's very helpful. Thanks,
Yes,
Lisa.
I will do that.
Mika will ask you.
Because I keep doing, it is strange, but you keep doing the same randomization. So you're definitely programmed to do it a certain way.
Well, I mean, I totally agree with what you said, Lisa. I think just having someone unbiased, not involved in the study, not knowing the treatments is probably a good way to do it. And even more so asking a biostatistician, biometrician. Maybe just a last opportunity for anyone from the floor to ask questions before we end off the session. Anything else? There's someone joining now at the very end. Okay, I'll let them in. Anyway, but with that, I want to say thanks a lot for everyone who attended this Soil Health Forum. I know the topic was not specifically on soil health, but the idea was that we need to have the foundation solid when we're doing work related in the field or in the glasshouse. And that obviously includes when we're looking at soil health questions as well. So thanks to anyone. Liesl, if it's okay with you as well, I'll share the PDF slides then with everyone along with the recording. And if there are any follow-up questions, I think the presenters made it clear that we are free to contact them. The information was shared before, but anyone's welcome to contact me as well, and I can share more information. So with that, again, thank you very much for everyone's time, and we will hopefully have this forum meeting again later this year.
Thank you.
Thanks, everyone.
Thank you very much. Thanks. Bye-bye.